Okay, good morning, everyone. Good morning, everyone. Yeah, this is better. So who of you has a jet lag from the 1 hour time difference? Who of you has not slept well tonight? Who of you just took a cup of coffee to make sure that you are awake this morning? So it's not very popular to drink coffee early in Lisbon. So I'm asking these questions because I think the first and important learning point is that, as we said yesterday, this technique is turning MRI into a measurement device. So it means that all the physiology will affect our measurement. So whether the subject has slept well, whether he's drinking some alcohol the night before, and also whether the subject had some caffeine, those things will all change the physiology and will change what we are measuring. So a nice first question is always, that's why the cup of coffee is here. I just got a question why the cup of coffee was there already yesterday. So think about what a cup of coffee will do to your perfusion. So what you can do if you want to test this is you give 200 milligrams of caffeine, that you can do it with a pill. It's as similar as 2.5 or 3 espressos. And then after half an hour on average, depending on the uptake, you get the maximum effect. And you can very clearly see that on your perfusion weighted image, on your arterial spin labeling image. So think about what happens with coffee. So who of you thinks that when you drink coffee, that your perfusion will go up? So remember the neurovascular coupling. So our brain starts working more, the neurons start taking more energy when we drink coffee. Half an hour after the cup of coffee, that's the maximum effect. So there's a higher demand for blood flow, right? So who of you thinks that the half an hour after drinking coffee, your perfusion goes up? Yeah, so if you repeat the question, there are coming some hands. Who of you thinks that perfusion goes down? Okay. Who of you thinks that the perfusion stays the same? Okay, some people need to wake up still in order to join this. Yeah, so or they are trying to figure out the Wi-Fi. So if you trying to figure out the Wi-Fi, easiest is to join the guest Wi-Fi. It's a guest IM whatever a few letters. And the alternative is Ederom also works, but the guest you don't need to sign into anything. So the answer is that half an hour after getting caffeine, your perfusion goes down. And that is, remember from yesterday, we actually are sensitive to vascular effects, to the supply of blood flow and to the demand of blood flow. And the vascular effect is stronger. So the coffee also acts on vascular constriction caffeine receptors, and that will give a vasoconstriction. So the perfusion goes down with about 20%. And we have in our brain, we have something called the oxygen extraction fraction. So even if the perfusion goes down with 20%, we can still take up more oxygen from the flowing blood so that we don't suffer from slightly lower perfusion. So remember that we always measure something vascular and we always measure so the supply of blood flow, and we are also sensitive to the demand of blood flow. And that physiology is very important. So these are the learning points. And I will go by them one by one this morning. It's a quite difficult start. So pay attention. Also, at the end of the day, we have a quiz where you can win a very nice local prize. Ta-da! So pay attention. So this is a paper that we did a while ago from Patricia Clément from Belgium. There were about 500 papers she could find with perfusion modifiers, and she nicely put them all together here. So the biggest are, as I said, coffee, nicotine, and other similar drugs, and also the time of day can change. Also, if you somebody didn't drink in the morning, osmolality will be, then the hematocrit will be very different. Hematocrit is an important factor, as we will see. So these are all things to take into account, and the community hasn't decided on whether we had. So typically you could say 2 hours before the scan, you cannot drink coffee, but that also will have a compensatory hyperperfusion effect. And so some say we should do that, and some say that we should actually keep the subjects doing what they normally do, scan them in their normal sort of resting state, and try to account for this. But this is not decided yet. But what it will do is, if you within the same scan session repeat your ASL, and this is the same if you do water PET. So in that sense, this is the physiological variability is on the whole brain level is higher than the technical variability has, so the low SNR, as we've seen. And if you repeat later on, you will get this as a 10% within subject coefficient of variability. So that means that the standard deviation over time is about 10%. And so if your perfusion is about 50 milligrams per 100 gram per minute, then that will be a 5% difference. And between subjects, it's about double that, which is quite large if you think that some dementia effects can be like a few percent. But as we saw yesterday, with tumors and with vascular effects, large effects are usually larger than this or more local. So that is important to take into account. Another big effect is if you just had some food. Who of you had breakfast this morning? Okay, so that is a very Portugal thing to have breakfast. The French, I think, have a very small breakfast. Our breakfast was really nice this morning. So who of you thinks that your perfusion increases after breakfast? You still have to wake up a bit, I think. Who of you thinks that your perfusion goes down after breakfast? Yeah. Okay, no postprandial depression. I mean, I don't see very depressed people here. Who of you think that after the food intake, your perfusion stays the same? So the digestion gives some CO2 as a result, and CO2 is a very potent vasodilator. So coffee constricts the vessels to the brain, and CO2 dilates it. And again, we have all these compensatory mechanisms, but this is something you can easily see. And it's also sometimes used as Dr. Vilela showed yesterday, you can use it as a challenge. As if you want to induce changes in blood flow, you can scan somebody, and then after 30 minutes with caffeine, or you scan somebody and you give them CO2. Usually this is air that is enriched with 5% CO2, and you see that the perfusion will increase. And this is used sometimes as a challenge to see if the perfusion can actually still decrease, if the vessels can still dilate. Okay, so we've talked about this yesterday. So small repetition, we do the labeling, which is jargon for changing the magnetization. And the only things that will flow up are the arterial spins. Spins are jargon for water protons. So we're looking at the water in the blood. This is our endogenous tracer. So it's as if we inject the tracer in the carotids and in the vertebral arteries. And then after this waiting time, which is very important, we acquire this image. In reality, if we would only acquire the labeled image here, what we will have is we will have water from the brain. Remember that the majority of water in the brain is from the stationary tissue, and only a few percent is from the blood. What we do then to not see this huge tissue effect is what we do is we also acquire a control image, which has only water from the tissue. It also has water from the blood, but that hasn't changed. So that means that if you subtract those, the tissue is the same, so that becomes 0. And in the first control condition, the blood was positive, and in the second label condition, the blood was negative because we changed the magnetization. So it's 1 minus 1. So you get 12 twice the perfusion. And we account for this in the quantification. And as you see below, this is very hard to detect visually because, again, the majority is water in the tissue. And if you subtract it, you see this perfusion contrast that I explained yesterday between gray white matter. But it's very subtle, it's very noisy because, again, the water in the blood is very low compared to the total brain. The cerebral blood volume is quite low, so about a few percent. So what do? We average it 30 times. This is an example of 2D epi. So 2D, the classical ASL scans were 2D echoplanar imaging. And then you do this, and it takes about 5 minutes. So this is 4 seconds, 4 seconds. And so that means that one acquisition is 8 seconds, and you repeat this 30 times. And then today we have more 3D acquisitions that take longer and have a different setup. So why is this not perfect? Life is never perfect. So again, what I explained, if the patient has exactly, or the control has exactly the same tissue on the first and the second image, and a control and a label image, that will subtract until 0. But of course, in reality, what happens is that the brain moves, and especially patients might move more than controls. So what you see is that if you move, especially it's easy to see on the edges where you have air. So in one case there's air, so there's 0, and in the other case somebody moved, so there's a high signal. So the subtraction, what it will give is this, you will see that the gray and white matter contrast becomes more blurrier, but it's especially easy to notice on this rim, because air minus a lot of signal, you get this huge motion artifact. So the rims make it easier to see. And what we have today is something called background suppression. So what that does is it makes sure that the tissue signal, so the water in the tissue, has a very low signal. So it suppresses the background, the tissue, so that subtraction artifact is lower. So you see with background suppression on, you don't see these rims, and there's still good gray and white matter contrast. But if the background suppression is off with earlier techniques, you can sometimes see these. So this is a important motion artifact to remember for yourselves, but also for the quiz. And without any motion, it's of course a perfect image. And sometimes this is called pseudo-perfusion because we assume that everything in this subtraction control minus label is perfusion, but that's not always the case. Another pseudo-perfusion might be pulsation in the ventricles. So sometimes you see some signal in the ventricles, or you see in the eyes some movement, or you see perfusion in the nose. Those are other things that you can see. What we typically talk about is the pseudo continuous ASL. That's the sort of standard technique to use. What people started with, as Dr. Villela said yesterday, about 30 years ago, was pulsed ASL. And the difference is in the labeling strategy. So what pulsed ASL does is it very quickly, and you see this on this scheme on the top, that you see that in time here, it's a very small in time, a very short pulse, which labels, because spatially it's very wide, it's like a slab. So it labels the entire, let's say, 8 centimeters of the feeding vessels. So all the blood that goes through there is then labeled. But the tricky thing is that you cannot time it easily. So they typically have, they end up in relatively short PLDs. So it's tricky to get rid of the vascular effect. And also the signal to noise ratio is lower. So what we got is that people thought, well, to increase the signal to noise ratio, let's label with a thin slab, but we make it wider in time. So we label for 2 seconds. So what then happens is that all the blood that goes through the thin slab, but according to 2 seconds, is labeled. So the signal to noise ratio is higher because you can label more. And also you have fewer vascular artifacts because you have sort of, if you would put this in time, the labeling duration would be around 0.7 or 0.8 seconds. Whereas if you put this in time, then usually the recommendation is about 1.8 or 2 seconds. So you have a much wider labeling duration, so more SNR, and also the bolus is more spread in time. So this bolus is more very fixed, very short. So you see more vascular artifacts, but it's easier to follow it. Whereas this bolus, it disperses more, but you see fewer vascular artifacts, and it's a better SNR. The disadvantage of this is that this labeling continuously gives you a very high SAR, so specific absorption rate. So the trick then is to do this pseudo-continuous. So what they did instead is they do very rapidly, they do these pulses that has the same effect as continuously labeling, but with lower SAR. So that's why usually people today, we have pseudo-continuous ASL on our scanners. And in 2005, there was this, 2014, there was this white paper. The sort of all the ASL gurus came together and said, okay, what should be the technique that everyone should use in the clinics? And that's when they said it should be P-Castle, it should be the background suppression that we just discussed. Another thing they said is that we should have a reference image for quantification. That reference image is called the M0. So the zero magnetization, which says, okay, with normal tissue water, normal blood water, what is the reference arbitrary unit of our MRI scanner so that we can quantify it. So what you end up doing is we have this subtraction, control minus label, and you divide by this reference scan, by this reference M0. So those are the three things that they recommended in that white paper. So pseudo-continuous ASL, background suppression, and M0. And the fourth thing is that they recommended the 3D ASL acquisition, which I'll also show some images from later. The first challenge, this labeling efficiency that I showed you yesterday, also interacts with the physiology of the patient. So if you look at this image, what you see is that on the radiological left, you see that the artery is straight, and we need to be perpendicular with our labeling plane to the artery to get a very efficient labeling. And so the more, if it would be completely straight, the labeling efficiency would be parallel would be 0. And if it's perpendicular, it's 100%. But what happened at the other side is that you see it got labeled, but then it got labeled negatively because you have this curve. So that's why it looks like we have a subject with very low CBF at one side. But in reality, this was just a labeling asymmetry. So that's another thing that might come back also in the quiz, to look at labeling asymmetries. Because usually if this is a reality perfusion scan, then you would have seen a hemiplegic patient, so you wouldn't need to have done the ASL scan. And depending on the vendor, so the biggest vendors are GE, Philips, and Siemens that we have a lot of ASL scans on, and the newer vendors like Canon and United Imaging are also implementing ASL. But you can have these three sort of flavors. So the poorest flavor of your labeling means that the labeling plane is 9 centimeters from the center of the imaging box, and it's fixed. So you cannot change the imaging box, and you cannot change the labeling plane. This 9 centimeters is what also was in the white paper, so that is usually fine. But if it's fixed, you cannot. So what the recommendation is that you make this quick angiogram with time of flight or phase contrast, and usually that's half a minute. You do this coronal and sagittal, and then you can try to tweak the labeling plane. But the poor setting is that you cannot tweak it at all. And then slightly better is that still the labeling plane is fixed to your imaging box, the imaging slab, but you can rotate your imaging box, and by that you take the labeling plane along. And the best is that you can tweak them independently. And some sequences you have to enable that. So ask your physicist if this is possible. And what I've seen is that if you train your technicians, they are quite able to look at the vessels and to do a nice positioning. So you want to be above the bifurcation, and you want to be below the siphon, and also try to avoid where the vertebrae the vertebral arteries exit the vertebrae, because they also make this curve. So usually it's a bit trickier on the back than on the front. And also, as you know, there's a huge aging effect. If we are young, we have nice straight vessels, so you won't see a lot of labeling issues. But if with older people, you get more tortuous vessels, so it becomes more difficult to nicely plan the labeling plane, and it's easier to see labeling asymmetries. We already seen this yesterday. This timing is very important, because if we wait too long, the signal has decayed. And if we're too early, then it's still in the feeding vessels. And so we always have some form of combination of an angiogram and a late perfusion phase. And traditionally, everyone wanted the perfusion phase, didn't look at the angiogram, but more and more we are seeing that this also provides us with information. And so both the vascular and the tissue CBF demand, that both of gives us information. We need a little bit of anatomy for that, so I'll skip through that quickly. And so again, you see that in the back, it's more difficult to get a straight labeling plane. And in the front, you want to be above the bifurcation and certainly below the siphon. And then we have what we usually can see also in those previous images. You have the anterior circulation, the anterior carotid artery, cerebral artery, the posterior cerebral artery, and also from the front, we have the middle cerebral artery. And if you haven't waited long enough, you will see that in the watershed areas, and especially in the part where the ACA, MCA, and PCA join at the back, you will see that the blood may not have arrived there yet, which will also come back into the quiz. As Dr. Vilela showed, you can also try to label only a single vessel, which they call regional perfusion imaging. It's not used a lot, but it can be very useful in vascular diseases. And then you see these types of images. So for example, what you can see is this is before a carotid endarterectomy, and below it's after. So you see that because blue and green and yellow, green and red here are the colors from the three labels the vessels that were separately labeled. And you see that after the surgery, the pressure difference is restored. So you see a nicer division between the left and the right intercranial, intercarotid artery. So you also see that the colorization is not needed anymore. And as we said already, the white matter blood flow is lower, and it also takes longer to get there. So this is why white matter ASL is quite tricky. People are starting to do it, but it is more tricky. And again, the timing is very important in this physiological technique. Sometimes instead of giving you what I would recommend in clinics, giving you an early phase scan, so that you see the vascular configuration, and also if there is any delay, in a late phase for perfusion. But sometimes they give you these arterial transit time maps. And I think it's nice, but it's not as helpful maybe as an early vascular phase. What you see here is how long for each voxel in the brain, volume element in the brain, how long did it take for the blood to get there. And you see nicely the watershed areas, and you see also that it takes the longest on the back top of the brain for the blood to arrive. And these are averages across a lot of subjects. And you can do this, for example, before acetazolamide, which is also a strong vasodilator, and after acetazolamide. So you see the same pattern, but you see that after the acetazolamide, the flow doesn't only go up, but also it arrives there faster. So you see that the arrival time in general goes down. So we can use this fact that we always have a vascular signal and a perfusion signal. We can use that also to do a quality control. So if you look at these images, what you see is a nice, well, as I explained, a nice contrast between the gray and the white matter. That's the first thing you want to see in a good quality ASL scan. So that means that the gray matter itself looks quite homogeneous. Then if the quality degrades, especially with vascular artifacts, but also with some head motion, what you see is that this contrast between gray and white matter becomes lower, becomes more difficult to see, and you start to see more vascular artifacts. And of course, if they really only have head motion, or the labeling went wrong, or there's a complete labeling asymmetry, then you start to see nonsense. So these are usually the three categories that we use. Either you see a nice tissue perfusion contrast, or it starts to be more vascular, which is the second category, or it becomes unusable, which would be the third category. And also, if you do this categorization on a large group level, what you see is that as it starts to become lower quality, and this is what we investigated, because usually, as I said, people threw these images away, but lower quality actually means more vascular signal. And as I said yesterday, we started investigating that, and the subjects with vascular signal actually also had more white matter lesions, were older, were more male, and had other risks of cardiovascular disease. And then if you take an average image of the unusual images, usable images, then of course it becomes trickier. And we can actually try to quantify this. So instead of looking at the mean perfusion in the gray matter, what you can do is look at the spread of the signal. And so if you look at the mean gray matter, let's say the whole gray matter across the whole brain, then the mean here is the same as the mean here. So that's important to note. The reason for that is that in both cases you had the same or similar labeling efficiency, so the same amount of blood arrived in the brain, but in one case it's all nicely in the tissue, so homogeneous, but in the other case it's the same amount of blood, but it's still in the feeding vessels. And more distally, the values are 0 because it didn't arrive there yet, because we didn't wait long enough. So the mean perfusion might be the same. So if you're interested in that, you could still use the image. But what you see is if you look at the standard deviation across the gray matter and divide it by the mean as a sort of normalization, then you see that the spread is obviously higher if you see more vascular feeding vessel patterns than that it has arrived in the tissue. And this is another way to quantify that, which you can visually see as arterial transit artifacts, but you can then you can also quantify that. I get a signal that I have to hurry up a bit. So timing is not important only in ASL, but also in giving lectures. So again, remember the labeling asymmetry. That's not a hemiplegic subject, but actually a labeling issue. This will come back into the quiz. Also, as I said yesterday, the blood T1 decay is dependent on hematocrit. So think about that always. The cerebral blood volume is the reason for us to scan longer and to have quite low resolution. And also remember the venous outflow, which you usually don't see, but in some patients or children, you might actually see that. I have explained this yesterday already. So an old male with type 2 diabetes mellitus has a high hematocrit. A young female with sickle cell anemia has a very low hematocrit. With a high hematocrit, the blood T1 is shorter, so the decay is faster, and we will underestimate CBF if we don't correct for that. With a low hematocrit, the signal survives longer, so the blood T1 is longer, and we will overestimate CBF if we don't account for that. So again, the venous outflow, sometimes you see it, and we don't account for that in quantification, but actually we saw that this can also be exactly as the arterial transit artifacts, also the venous artifacts can maybe give us a clue that there is some shunting in, for example, sickle cell disease patients, and also in some tumors you see that there is a shunt, and you see more vessels. So we can also, in that case, it's not really measuring CBF as good as it can, but you get extra information from the vascular, the arterial or venous side. So that's important to understand. I've shown this yesterday as well, but what is important to note is that you have both 2D ASL techniques, and more people are doing 3D ASL techniques. And what can be the side effect of the 3D, especially, so this is the same GE 3D ASL scan. This was the one that the physicist David Alsop created. And what you see is that with 2D, you have, like with fMRI and DTI, you have susceptibility artifacts in the front of the head near the air cavities and also near the mastoid. So with 3D ASL, we don't have that because it's spin echo weighted usually, and we have, especially with 3D spiral, have better whole brain coverage and higher SNR. But what sometimes they do, this is the same technique, but this is the clinical implementation. So the product sequence is that they smooth it a lot because it starts to look better to the radiologist. But also for research, but also I think clinically, this one is much nicer than that one. So that is possible. But when you see these 3D ASL techniques, they can be very blurry. And there are ways to deal with that and to make them less blurry, but this is all in investigation. So remember that roughly 2D techniques, they have more susceptibility artifacts. They have also slightly distortion, as you see with fMRI and DTI, which are similar 2D techniques. And with 3D spiral, which is spin echo weighted, you don't have this distortion, but it becomes more smooth, more blurry. And 3D grace is sort of in the middle. This is what the vendors used to have. Now both Siemens and Philips have 3D grace, and GE still uses 3D spiral. So this will also come back in all these things that I'm showing you will also lead to harmonization differences, which will also come back in the brain age talks. This is another way to look at it. If you have a lot of subjects, you can create average images. So you see already that this mean control image or the M0 image can be blurrier in 3D techniques. So that is important for registration. And so it's very difficult to register something that has a very blurry image. And then, as I'll show you later as well, even with blurrier techniques, you still have a nice gray white matter contrast. So that means for registration, sometimes the perfusion weighted image might be easier because the contrast is much higher in the perfusion weighted image than in the raw ASL, the unsubtracted ASL or the M0 image. The contrast is much lower. And these are the contrast that you see is usually T2 weighted, but with a much lower resolution than you're used to. And these are averages across a large sample size, so they look much nicer. But you can easily see also that with the 3D techniques, it's also good to understand that even though they report very low voxel sizes, so they say that they have a very high resolution, because of the blurring of the technique, it ends up to be still very blurry, and 2D acquisitions can still have a higher spatial resolution, as you can see here. That's it. So again, physiology is very important. We discussed the subtraction and potential head motion artifacts, for which we have background suppression, but still look at the rim edge. Labeling efficiency, we've discussed that. Look especially at the symmetries, and ask your physicist to make a quick scout of the vessels, coronal and sagittal, that you can plan this nicely. Look at arterial transit artifacts, but also in the venous side, and maybe that gives you extra information. We discussed the blood T1, and we discussed readout differences, and there'll be coming more when I will discuss the post-processing, how to deal with all this. Thanks.
Hi, thanks. Yeah, so I'm happy to see all the faces. So that means that you were not discouraged when we told you that there will be a lot of hands-on with computers. I'm also very happy to see all the computers. So I'm a computer scientist, so I'm more comfortable with computers than with faces. But yeah, I'm really happy to see the computers, which means that you have all the means that you really need to complete successfully the workshop. So first, I'll start a little bit with introductory lecture to give you motivation why you will have to endure all the stuff with the computers and all the just all the work with the code and all the unpleasant work. So yeah, in this educational talk, I'll try to explain to you what are the issues with storing and sharing ASL datasets, which go a bit beyond what is typical with normal medical datasets. So I guess many of you, when I was going through the list of registration, there are neuroradiologists, radiographers, scientists. So you're all probably very familiar with clinical data management, how this works. So you know that you need to store it securely using a nice paid service. Sometimes you need to anonymize the data. You need to talk with the patients. Yeah, they need to give consent to obtain the data, share the data. So these are all the things you're very familiar with, I suppose. But quantitative MRI, like ASL, it can bring some extra challenges that extend beyond what is standard for medical data. Um, so these challenges are that actually we say ASL, but it's not just ASL. It's a variety of keywords and concepts and sequences that are behind it. It can be velocities, selective ASL, pulsed ASL, pseudo-continuous ASL. So there's just a lot behind. Sometimes it's a big mess. And also, ASL comes with a variety of parameters. And to be able to properly quantify the data, to obtain the real quantitative values, not just to check it visually, you need to know about all these parameters. So all these parameters we've heard in the previous talk, like the labeling delay, labeling duration, all these play a significant role. If you don't have them right, if you don't have the correct name of the sequence, if you don't have the correct parameters, then the numbers you're getting out are just not realistic. So that's why you really need to be very careful about how you store the data, if you store it correctly, if you have all the parameters saved. Otherwise, you're not getting a proper quantitative output. So this is what we really need. We need to have everything properly stored. But what is the reality? The reality is that this is not usually happening. And I'm not pointing fingers to any of you because I believe you're doing your job correctly. But very often not the case. So very often the responsible researchers, they store the data and they start a clean version, and then they start a clean version update, clean version 2. They have it over several disks. There's no order. The data are missing. It's usually just a huge mess. And the problem is that very often it's a single person that knows how the data are stored correctly, and that single person the contract runs out, the person leaves for a job in industry or just disappears from academia, and there's no reasonable way how to recover all this. So the point here is that you want to, from the start point, store your data correctly and nicely, that none of these problems occur. And even if it's stored nicely, even if the researcher or the person responsible did a good job, then probably in a different center, they do it a completely other way. So if you start exchanging data, sending data to different people, then you still don't understand it because they use a different concept. So it's hard to understand it, or it's hard to write a software that can automate the processing because you have to adjust everything. So order is important. And you would think that, oh, but we have DICOM. DICOM is a standard. It has everything saved. All the parameters are nicely stored, but that's actually not completely true. It might be true for some basic MRI sequences, but when it comes to ASL, there are different vendors. They store all the parameters like PLD in different fields, or they store it in private fields, or they don't store it at all. So it's usually a huge mess, and it's very difficult to find out which parameters are really stored where, unless you have a very good information about the sequence. And as a person that is not very deeply into ASL for many years, you usually don't have it, and it's usually very difficult to even Google that and find that out. So these are all motivations why we need a very proper, good standard to store the data so that any person from any lab can just open the data and know what's really stored there nicely, understandably. So luckily, there starts to be certain solution to address this problem. So there's this open science initiative for perfusion imaging. These are people that are trying to put all the standards together, work together, and enforce some standards. So for example, now there is a ASL lexicon that lists all the key terms in ASL, explain what they exactly mean and what they really should mean. And they also give some standards on how to report ASL in scientific papers. And there is this BITS initiative, Brain Image Data Structure. So this is a way how to store and share MRI data, not just ASL. And that will be the key focus of the roughly next hour. So BITS is a very large initiative that is not only dealing with ASL data or not even with MRI data. It tries to provide some standards for MEG, EEG, and many other different sequences. So it was not created by computer scientists or data people, but by psychologists. So there might be some shortcomings, but yeah, no standard is perfect. So when we come to ASL, the basic concept is that we have kind of three kinds of data. There's the source data. This is the big mess that you're putting in. Could have any format, can be anything. This is just how you get your data from your scanner. Then you have your raw data. This is the clean data in standardized format, nice, standardized, saved how they should be. And then there's the derived data. This is when you take your nicely, cleanly stored data and you start processing it, calculating CBF maps, and so on. To roll back a little bit, give a little bit more layman explanation. There's a popular explanation to explain bits with a pasta example. So what I've been talking about, this huge mess of completely unsorted data, this is this first example. So if you want to eat pasta or share pasta, you definitely don't want to have this mess because that's just ugly. So as I was saying, the source data, this is something that's relatively nice coming out of the scanner. This is how you get stuff out of the shop. Then you want to neatly sort it, create it in a standardized form, cut it form, how everybody can work with that. This is the raw data. And this is what you can share, you can work with. And then you end up with the derived data. This is the fully finished food. This is how you do your analysis and work with it and use the data. So to give you an idea, how does it practically look? So the source data, how you download it from the scanner. This is just a bunch of DICOMs with some subject names, different subjects, different sequences acquired. Nobody can understand this except for you, because yeah, there's not really a structure, but it's not a mess. From that, you start creating several things. So you create your study named ASL4. you give it some descriptions, then you extract your subject name. This subject will have T1 weighted scans here in a Nifty format in the folder called anatomy, and it will have a so-called JSON sidecar. JSON, it's just a text file that has pairs of a name of a parameter and its value. It's human readable and editable, simple format. Then you'll have your perfusion scans in the folder perf. So there'll be the M0 calibration scan. You can have a JPEG that shows a snapshot of the labeling plane that you can just take with a camera on the scanner. And then you will finally have your ASL scan. So as with the other sequences, you will have the Nifty file that contains data, and you will have the JSON sidecar that contains very specific predefined parameters. So you can see this is exactly how it looks, fully understandable to everybody, human readable. So you have the magnetic field strength, 3 Tesla, pulse sequence type, it's EPI, acquisition, it's a 2D acquisition, echo time, it's a Picasso labeling, it has several post-labeling delays, background suppression is true, and so on and so on. So you can see that this is neatly organized. Any researcher can work with that, understand that, and yeah, and everybody has to, if the person wants to use the bits format, should save it in exactly this format. So this is our ideal outcome. Yeah, besides that, you should also provide information about which of your scans are controls, which are the labeled ones in a separate file. Yeah, so this all is the bits format. So the bits format doesn't do anything very difficult, anything very magical, just describes that you should name your subjects inside GIF folders with anatomical scan, perfusion scan. They should have a text file attached to it that has this specific tags. That's all. Okay, so added value of, obviously, if you just work on your single data set in your lab, then you might be happy with your messy data set because you know what's there, you know how to work with it. So, and it requires extra effort to convert it to a standardized form because you need to learn a bit about the standard and convert the data. But it comes with advantages. So once you do this basic work, it's you can use pipelines that are compatible with that. So for example, if you have data in bits, you can directly feed it to Explorer ASL pipeline. You don't have to do anything special. The pipeline automatically recognize how the data look like, what parameters are there. You don't have to configure it, you just process it. There starts to be more pipelines that work with ASL data as well. When it comes to other MRI data, there are much more software packages available. For ASL is just starting, but nevertheless, hopefully in few years, you just put data in correct format, you choose the pipeline, and the pipeline will be able to work with it without any extra settings. The other advantages is that BITS starts to be a large community addressing several sequences. So there are more packages, Python packages, MATLAB packages to read the data, organize the data. There are some online manuals how to start working with it. There's a validator, which means, again, a big advantage. If you organize your data set in the bits format, You can upload it to the validator, and the validator will confirm that the data is complete and without errors. So imagine that you're beginner in the ASL field. You put your data, you put all the parameters, but you forget the post-labeling delay, which is an important parameter, but you might not know that. So if you just upload it to this web page, the validator will tell you you're missing post-labeling delay. You have to fill this in, and you can find this immediately and not after 1 year when somebody starts to playing with your data and tells you that you've been missing this parameter all along. Another advantage is a tool that we've developed together with OCP, supported by Google Summer of Code, with the help of this talented student from Johns Hopkins. And this is automatic reporting. So if you have ASL data in bits format, you can just upload it to this web app, and it will generate the method sections for your article. So just uploading the data in standardized format, it will write you this text. Oops. Yeah, that will tell you, that will report it exactly in the format that the journals should accept with, yeah, you not necessarily knowing all the details about ASL. So to recap and to introduce again how this will work in Explore ASL. So we start with the source data. So source data, this is the usually DICOM data in the way how you want to have it. This is then converted to Nifties with DICOM to Nifty. It creates the Nifty data. Then in Explore SL, we go through all the Nifties, we check the DICOMs, we make sure that all parameters are extracted from the DICOMs, from the hidden tags, private tags, if they're there, or supplement extra parameters manually. And this then creates the raw data structure. This is the clean data containing all the parameters before processing, but in a standardized format that allows you to share it, store it, process it, do anything with it. And then after we have this raw data, you can process it with Explore SL or any other pipeline. You can do your work with it and obtain the derived data, which is the processed CBF maps used for analysis and research. So that's end of the theoretical talk. Perhaps if you have any questions now, it might be a good idea to ask just to make sure that you understand the concept of everything. Of course, there'll be much more time to ask when we really start working with it. So then the questions will pop up. So all good.
No questions. But either you did a very good explanation, or nobody
Well, we're ready for that. So once we start the practicals, I'll see all the wrinkles on your faces, and then I'll know where to go. So it's no concern. No concern. Yeah, can you help me out with the next one?
Does the moderator have a question?
You don't need to. Thanks.
Submission.
Yeah. Yeah. Okay, so this is a part where I really have no estimate of how much time we're going to take. So ideally, we take 2 minutes and then we go for a coffee break, or we take 5 minutes and we'll still go to coffee break and we'll try to finalize everything after the session. So we keep it flexible. Now it's the time to open your computers because we'll start working with them. So yeah, if for all the advanced If you have any questions, especially for later, not really for now, because now you can ask us. If you later want to work with ASL and you have questions, then we have a manual that should contain most answers without too much errors and stuff. So. Yep. So we are able to support you on any type of computer, Windows, Mac, Linux, that should all work. So basically we have several scenarios. Either you have your own notebook, you can install software and you have MATLAB license. That is the easiest scenario. If you don't have a MATLAB license, that's all good because you can install the compiled version, MATLAB runtime. So you can just download stuff for free from the internet. It will not cost you anything. This also works. If you don't want to install anything on your computer, then you can connect to our server where everything is pre-installed. So this option is also, if anybody doesn't manage to install stuff doesn't work, then you can come to us, connect to the server, and that's going to be easy. So just a quick survey. Who has a MATLAB license running on a computer? Wow, nice, nice for MathWorks. Who is able to download stuff on the computer and install? Okay, a lot of hands. And who has a computer, is willing to work with a computer but cannot install stuff and needs to connect to our server? Okay, so if I might ask Matthijs to hand over the password. Yeah. So so right now you can't do anything with it. You don't know where to connect, but we'll show that later. So I was asking you yesterday to please download software and data. I was also sending an email. You can go to the same link. That is the exact link I shared with you before. So yeah. I see their cell phones up, but okay. So I'll just go very quickly, explain the concept, then we'll discuss during the coffee break if you need to do more. So this is just going to be quickly, and I understand that you'll not manage to do it. So if you have MATLAB license installed, you just go to the GitHub pages, Explore ASL. You select branch develop here. And then if you have a GitHub client installed, you just download the develop branch, or you download the zip file, or you just follow the link I've sent by email. This downloads you the code. You should have the code. You unzip the code, and then you open MATLAB. That's just it. That should be pretty easy. Yeah. Once you have it unzipped, you just go to that folder and run this command, explore ASL initialize. So you go to the folder with the code, start MATLAB, run explore ASL initialized, and that should be it. You should obtain this screen and you're good to go. So I just assume that you have it, but after the coffee break, we'll go through this in a little bit slower way. If you want to use the compiled MATLAB version, then you need to download the MATLAB runtime environment from the links I've sent. You need to download the MATLAB compiled version called XASL Lisbon. You just have to make sure that you download the correct version 2019b from the MATLAB runtime, and you download the zip file from the correct folder. And then you execute in a slightly similar way on Windows. You just unzip the folder, write xasl latest.exe, and then empty string 00. Or on Linux or Mac, you write this xasl latest sh. You provide path to the runtime environment and the same. This should get you to the same folder. Now, Teresa. So we don't seem to be having the name of the server here. So we'll just, yeah. Since it's just Teresa today, I'll help you out to give you the name of the server, and then you go to the folder, you run the script, and you will be all good to go. So just a quick question. So did anybody in this very rapid session manage to install stuff? So whom of you managed to download the software and the data? One, a few. Anybody managed to actually run the code? No, not yet. Okay, yeah, it's all good. So we'll have a coffee break. We'll get some sweets, coffee, and after that we'll try to really execute everything. Okay, thank you.